Hello, today's subject is how to identify bacteria. This can be done in numerous different ways, and we have to keep in mind that the classical methods only really works on bacteria you can grow on an agar plate. So the first thing to do is to do a visual inspection. Look at the colonies. What is the shape? What is the color of the colony? Is it growing fast? Is it growing slow? In here you can also add smell. Very experienced technicians, lab technicians, they can smell a bacteria and say, this is Pseudomonas, this is E. coli. And it doesn't really take a lot of practice before you're able to do that. But when you're new, you still have limited experience, so that's one of the more difficult ones. The visual inspection, some of the materials you see in the book and in the exercise manual, there are some descriptions on how to see what a bacteria looks like or the colony looks like. So that's the first part. The second part is to use a microscope. And this can tell you shape. Is it a coccus? Is it a rod? Is it a filamentous something? It can tell you that. Size again. If you look at yeast compared to an E. coli, the yeast cell is huge compared to that. So size is also something you see. And then last and very important thing you can see in a microscope is movement. Is a bacteria lying still? Is it moving around? Again, a very important piece of information you can look up in your book. If you suspect it is this bacteria, okay, is it moving or not? Does it have flagella? Next thing is, of course, to do a gram identification. Is it gram positive? Is it gram negative? In this exercises you're going to do in this lab course, you're going to use a gram stain, a modification of the original gram stain, and you're also going to use the potassium hydroxide test. And both of them will, with reasonably clear, show you if it's a gram positive or gram negative. And this is very important identification, because this piece of information you can rule out at least 50% of the bacteria by saying is it gram positive or gram negative, or being able to determine if it's gram positive or gram negative. So when we've gone this far, you're actually very close at being able to say, at least have a pretty good idea about what bacteria it is. You can move into the biochemistry. You can check if there's any particular enzymes that this bacteria you suspect it is have or does not have. It could be if it has an enzyme like catalase. This enzyme makes this reaction happen. And this is very easy. You simply put a drop of hydrogen peroxide on the colony, and when it starts bubbling, it's because oxygen is released. So this is a very simple test. So you would be able to say this bacteria, is it catalase positive or negative? The next thing you can do is to move into the area of where you select. You have in this suspicion this bacteria is present. Now you would make circumstances so that you select for growth of this particular bacteria you suspect it is. This can be done, of course, with temperature. You can look up in the book what is the temperature where this particular bacteria will grow, pH, and of course if it's aerobic or anaerobic. So all of this can be done fairly simple and tell you a lot about it. But you can also move on to particular media which contains nutrients which only your particular bacteria is able to use, or media with some kind of indicator that will tell you if you have this particular color, it's because there is a bacteria growing there releasing enzymes that would change something in the media so that your plate simply becomes purple or red or whatever. And then if you've gone this far, you should be able to say exactly what kind of bacteria is present in your sample. Nowadays, a lot of bacterial identification is done using molecular techniques, and it's becoming more and more popular. One big advantage with molecular techniques is that you are able to find bacteria which will not grow on an agar plate, like the, how should I say, the classical identification looks into. Secondly, they're usually much faster. We talk about a couple of hours, and then you have not only what group of bacteria, but you could actually tell what kind of a strain it is, and even give an indication if it's a resistant strain or not. So this has become of vital importance in the field of clinical microbiology. That means microbiology you do when you want to set up a diagnosis for a patient in hospitals and so on. And we can basically split the molecular techniques up into two. Those where you use DNA and those where you use protein. And the DNA based, it can be PCR based or it can be sequencing based. Basically you can say in PCR based, then you know what you're looking for. You have a specific bacteria you want to find out, is it this or is it not this? Sequencing, you can throw out a little broader net and see what you catch. And they a couple of years ago, PCR invaded the clinical microbiology labs. Nowadays, sequencing is moving in. Protein, it is almost only this method called multi-TOF. It's a kind of mass spec. And basically what you do is you put the bacteria into this instrument. You might have crushed it, and you take a colony, whatever. And in a mass spec, to put it shortly, you simply add a charge, the same charge, to all molecules in the soup of your crushed bacteria. And then you apply a high charge, opposite charge of what the charge you added, and that will kick the proteins through a tube, and the end sets some kind of detection system. The smaller the molecule, the longer it will fly, and that will tell you. You get a specific pattern of different proteins and part of peptides, etc., and coupled to these multi-TOF systems are a big computer with a database, and it will say it's an E. coli, and it will give you the strain too. More about these techniques later in your live here at Epsilon. So this video was about how to identify bacteria, what you have to think of, and a workflow. So visual inspection, using a microscope, gram stain, biochemistry, and maybe a selector media and stuff like that. Here you see the cupboard in the lab where we have the different media and things for microbial growth. And on the top shelf we have a lot of different selective media which can be used to identify bacteria, because it will be possible for it to select for growth of only specific bacteria. We have the MC17 agar, which is used to detect lactic streptococci. We have something called McConkey agar, which is used to select for gram-negative bacteria. There's something in here that inhibits growth of gram-positive bacteria. We have the bile esculin acid agar, which will detect facial streptococci, meaning streptococcus from the faces, and a lot of other types of agar, which can be used and usually used when you have some kind of idea from the visual inspection, using the microscope, biochemistry and so on. Then you will turn to some of these, make some plates, plate out the bacteria you have an idea what it is, and see if you have growth or not growth. Both things will tell you something. A little about the selective media which is present. You can also make your media more selective by adding different types of nutrients like different types of sugar, etc.